Hello everyone, my name is Mehmet Tozoglu, and I'm a rising fourth year microbiology student. Probably just like the many of you here, I'm really excited to present my journal club topic today. The article I'll be discussing is the human gut microbiome and early onset type 1 diabetes. This topic is especially meaningful to me because I have a close family history of type 1 diabetes. Growing up around someone managing this condition sparked my interest in understanding not just how it develops, but also what might be done to detect or prevent it early. Type 1 diabetes is an autoimmune disease, and recent research has started to uncover connections between the immune system and the gut microbiome. So I was drawn to this paper because it explores whether scientific patterns in the gut microbiome might contribute to or even predict the onset of type 1 diabetes in children. I'm hoping that by understanding this relationship, we might someday improve early diagnosis and explore new preventative strategies for families like mine. With that being said, let's jump into the bottom line takeaway from this study, my bluff statement. Understanding how the gut microbiome differs in children who develop early onset type 1 diabetes compared to those who don't can lead to the discovery of early diagnostic markers and the development of novel therapeutic strategies. The study specifically identifies unique microbial patterns associated with type 1 diabetes, highlighting the gut microbiome's potential role in both the development and progression of the disease. Now, why is this important? For those who may not know, type 1 diabetes is a chronic autoimmune condition where the body's immune system mistakenly attacks the insulin producing beta cells in the pancreas. It primarily affects children and young adults, and its global incidence is steadily rising. This makes it critical to understand what triggers the disease in the first place and how we might prevent it. The gut microbiome, composed of trillions of microbes in our digestive tract, is known to play a central role in immune system development and regulation. By studying how the gut microbiome shifts over time, we may gain insight into early disease mechanisms and find ways to intervene before the immune system turns against the body. So the central research question of this study is, how does the gut microbiome composition differ in children who go on to develop type 1 diabetes compared to those who don't? That's what we're here to unpack. Now that we've laid out the main question, let's dive a bit deeper into some background information. The gut microbiome is made up of trillions of microorganisms, including bacteria, viruses, fungi, and other microbes that live in the digestive tract. These microbes play essential roles in digestion, nutrient absorption, and especially immune system regulation. When it comes to the immune system, the gut microbiome is critical for both its development and proper function, and it helps train the immune system to distinguish between harmful pathogens and harmless substances, which in turn helps reduce inappropriate immune responses like those seen in autoimmune diseases. When the microbial community becomes imbalanced, a condition called dysbiosis, it can contribute to immune dysregulation and inflammation. In fact, prior studies have shown that individuals with autoimmune conditions often have distinct gut microbiome profiles compared to healthy individuals. These differences can include reduced microbial diversity and changes in the abundance of specific bacterial taxa. Now, specifically in the case of type 1 diabetes, a cohort study from 2015 explored the gut microbiome in infants and found trends suggesting that a unique microbial composition may be associated with T1D development. However, the findings were inconsistent and the relationship remained unclear. One of the main issues is that many earlier studies only captured a snapshot of the microbiome at a single point in time, but since then the microbiome evolves, especially in infants. We need longitudinal studies which follow subjects over time to better understand how these microbial patterns develop and potentially contribute to disease. That's exactly the kind of study we'll be looking at in this presentation. So now you might be wondering, what is the need for this study? Well, according to a 2017 article from the New England Journal of Medicine, there is an annual 3 to 5% increase in the incidence of type 1 diabetes among children and adolescents. This is alarming because type 1 diabetes is a lifelong autoimmune disease that requires constant management, primarily through insulin therapy. Without proper treatment and monitoring, T1D can lead to serious complications like cardiovascular disease, kidney failure, and an overall reduced quality of life. It also significantly raises healthcare costs and can place an emotional and financial burden on families. That's why identifying specific gut microbiome patterns associated with the development of type 1 diabetes is so important. If we can pinpoint these patterns early, we may be able to create diagnostic markers to identify at-risk children before symptoms even appear. With early diagnosis comes the possibility for timely intervention, which could delay or even prevent the onset of the disease altogether. This would reduce the burden of the disease on both the individual and the healthcare system. Additionally, this research could lead to innovative microbiome-based therapies such as probiotics, prebiotics, or even personalized medicine tailored to someone's unique gut profile. Finally, gaining a deeper understanding of how the gut microbiome relates to the pathogenesis of T1D could not only help us treat this disease more effectively, but also expand our knowledge of other autoimmune diseases. Now that we've covered the background and the need for this research, let's dive into the actual article and what the study aimed to do in simple terms. The primary goal the primary goal of this study was to identify specific gut microbiome patterns that appear in children who go on to develop early onset type 1 diabetes compared to those who remain healthy. By analyzing and comparing the gut microbiota of both groups, the researchers hoped to uncover potential biomarkers, basically biological warning signs, that could be used for early diagnosis of the disease. In doing so, they also wanted to better understand the role of the microbiome in the actual development and progression of type 1 diabetes. So overall, the study isn't just about finding differences, it's about using those differences to learn how the microbiome might actually influence or even contribute to the disease process itself. So now that we have talked about the study's purpose, let's look into how it was actually carried out. To analyze the gut microbiome, the researchers used two main sequencing technologies. The first was 16S rRNA gene sequencing, which many of you might be familiar with from this class. For those who aren't, 16S rRNA sequencing is a method that allows researchers to identify and quantify bacterial species present in a sample, specifically stool samples in this case. It targets a conserved region of the bacterial genome, allowing for classification at multiple taxonomic levels. The second method they used was shotgun metagenomic sequencing. Unlike 16S sequencing, which focuses only on bacteria, shotgun sequencing captures all genetic material in a sample, including bacteria, viruses, and their microbiomes. This gives a broader picture and also allows researchers to explore the functional potential of the microbiomes, not just who's there, but what they might be doing metabolically. The data from this study came from a larger project known as the TEDdy study, which stands for the Environmental Determinants of Diabetes in the Young. This is a multinational cohort study designed to uncover environmental factors contributing to type 1 diabetes. The TEDdy cohort included children who were genetically at high risk for developing the disease, and samples were collected over time. Now that we've covered the study design, let's talk about how the samples were actually processed. The sample collection process involved gathering stool samples from children at multiple time points, starting in infancy and continuing into early childhood. This longitudinal approach was crucial because it allowed researchers to track how each child's gut microbiome changed over time, instead of just looking at it at a single snapshot. Once collected, the genomic DNA was extracted from each stool sample using standardized protocols. This consistency across all samples was important to make sure the data was reliable and could be compared across time and participants. After DNA extraction, researchers applied both 16S rRNA gene sequencing and shotgun metagenomic sequencing, the methods I described in the previous slide. These techniques help them gather data on not just which microbes were present, but also what those microbes might be doing, giving insight into their functional potential. Together, this approach gave researchers a powerful, high-resolution view of how the microbiome composition and function differed in children who went on to develop type 1 diabetes compared to those who didn't. Once the sequencing data was collected, the researchers needed to make sense of it, and that's where the data analysis comes in. To ensure reliable and meaningful results, the study used both comparative analysis and advanced statistical methods. The comparative analysis was used to directly compare the gut microbiome composition and functional gene profiles between children who developed type 1 diabetes and those who did not. This helped pinpoint specific microbial patterns that were consistently associated with the disease. In addition, the researchers used advanced statistics such as abundance testing and correlation analysis. These methods helped confirm whether the differences they were seeing were truly significant or just random noise. For example, they looked at how much of a certain bacteria was present and whether its abundance was statistically different between the two groups. Some of you may remember the concept of statistical significance from previous courses. Basically, it means the differences we observe are unlikely to be due to random chance. Altogether, this combination of methods gave researchers a robust and comprehensive framework of interpreting the gut microbiome's role in type 1 diabetes. Now we're getting into the heart of the presentation, the actual result of the study. The most important takeaway here is that there were clear and significant differences in the gut microbiome composition between the children who developed type 1 diabetes and those who did not. One of the standout findings was that the children who developed T1D had significantly lower microbial diversity. As I mentioned earlier, this is a classic sign of dysbiosis, which means there is an unhealthy imbalance in the microbial community. When the gut microbiome becomes less diverse, it can affect how well the immune system is trained and regulated. Looking closer at the specific changes in bacterial populations, the study found decreased levels of beneficial bacteria. These bacteria are known for their anti-inflammatory roles and for maintaining gut health. On the other hand, there was an increased presence of more pathogenic bacteria. These types of bacteria are often associated with inflammation and gut barrier dysfunction, which can contribute to autoimmune processes like those seen in type 1 diabetes. In short, less of the good guys, more of the potentially harmful ones, which may play a big role in disease development. Now that we've covered the key findings, let's dive into the visuals that really help us understand the data. Starting with figure 1 on the left, this diagram maps out the timeline of sample collection in relation to key events in disease progression. The study followed children from infancy to early childhood, and the figure helps us see how their microbiome changed over time. You'll notice healthy controls are shown as circles representing consistent samples from children who never developed T1D. Individuals of those who developed autoantibodies indicating immune activation but not necessarily diabetes are shown with a transition from circles to diamonds, meaning the immune system has reacted to a trigger like a virus, and antibodies are now detachable in the blood. Lastly, children diagnosed with T1D are represented as squares, and their samples include data collected up to 1 month before diagnosis. This timeline emphasizes the longitudinal nature of the study, which is important because it tracks how the microbiome evolves rather than just capturing a snapshot. Now, if we move to the panel onto the right, we're looking at a Shannon Diversity Index plot, which measures the richness and evenness of microbial species. Each dot represents a sample's microbial diversity. Blue colors indicate lower diversity, red colors indicate higher diversity. You can clearly see that healthy controls maintain high diversity throughout, and seroconverted individuals show a slight dip, and children with T1D show a noticeable and consistent drop in diversity. This pattern supports the idea that gut dysbiosis gradually worsens as children move from healthy to seroconverted to T1D. It also reinforces the idea that microbiome changes aren't just a consequence of disease, they may actually precede and contribute to it. This figure takes us deeper into the functional role of the gut microbiome by showing how specific metabolic pathways and short-chain fatty acid production differ between children who develop type 1 diabetes and those who don't. We're looking at a heat map here which illustrates changes in the abundance of key enzyme families from birth through the first 2 years of life. The goal is to capture how the metabolic potential of the microbiome evolves over time. Red colors on the heat map indicate enzymes that are increasing in abundance. Blue show the enzymes that are decreasing. What stands out is that in the T1D group, there is a noticeable reduction in enzymes responsible for producing short-chain fatty acids, which are known to play a protective role in the gut health and immune regulation. These short-chain fatty acids, such as butyrate and acetate, are typically synthesized by beneficial gut microbiomes and help maintain intestinal barrier function and suppress inflammation. Some of the enzymes families that show decline in the T1D group include inorganic diphosphates, RNA nucleotide transferases, and NADP transhydrogenases. These are all involved in essential beneficial metabolic pathways. Their decline suggests that children developing T1D experience a functional dysbiosis, not just fewer good microbes, but fewer helpful functions being performed. At the bottom of the heat map, you'll also see the top 15 bacterial species that contribute most to these changes. These include beneficial microbes such as Bifidobacterium and Lactobacillus, which are significantly reduced in the T1D group. This functional shift supports the idea that gut dysbiosis in T1D is not only compositional but also metabolic, impacting the host's immune system and potentially playing a role in disease onset. This slide presents another heat map, but instead of enzyme activity, this one focuses on gene prevalence and abundance in the gut microbiome during breastfeeding and after weaning in children with and without type 1 diabetes. On the left, shades of green show gene prevalence, where darker greens means higher prevalence. On the right, shades of blue to red represents gene abundance, with blue indicating low and red indicating high. This figure specifically highlights Bifidobacterium longum, a beneficial gut bacterium that is commonly found in breastfed infants and is known for its probiotic properties. During the breastfeeding phase, certain genes are both highly prevalent and abundant. These genes support the infant's ability to digest breast milk efficiently, reinforcing the role of the gut microbiome in early nutrition. However, once weaning begins, meaning the infant transitions from breast milk to solid foods, there is a noticeable decline in both the prevalence and abundance of these genes. This can be seen especially with B7-GMP2 and B7-GMQ8, both of which drop significantly after breastfeeding ends. These patterns emphasize how the gut microbiome functionally adapts to dietary changes. In children who develop T1D, these functional shifts may occur differently or earlier, contributing to immune dysregulation. Overall, this figure reinforces the dynamic nature of the microbiome during critical developmental stages and its potential role in influencing disease risk. Beyond changes in which bacterial species are present, also called compositional dysbiosis, the study uncovered evidence of something called functional dysbiosis in the gut microbiomes of children who develop type 1 diabetes. So what does this mean? Well, functional dysbiosis refers to a shift not just in the types of bacteria but also in the metabolic activities these bacteria perform. In other words, even if the bacteria look similar, what they're doing inside the gut is very different in T1D cases. For example, the study found a reduction in short-chain fatty acid production, which is crucial for maintaining a strong and healthy gut barrier. When short-chain fatty acids are lacking, the gut lining becomes more permeable, often referred to as a leaky gut. This increased permeability allows harmful substances to cross into the bloodstream, triggering inflammation and potentially contributing to the autoimmune attack that destroys insulin-producing cells in the pancreas. This tells us that the gut microbiome isn't just a passive passenger, it's playing an active role in how the disease develops. The exciting part is that this creates an opportunity. If we can restore beneficial bacteria and improve SCFA production through interventions like diet, probiotics, or microbiome-targeted therapies, we might be able to delay or even prevent the onset of type 1 diabetes. In addition, the specific microbial and metabolic patterns identified here could serve as early biomarkers, allowing for earlier diagnosis and personalized preventive care in children at risk. Now that we've gone through the results, let's move into the discussion portion of the study. This study successfully identifies distinct microbial patterns associated with T1D, highlighting how the gut microbiome may play a crucial role in the development and progression of the disease. One of the biggest strengths of the study is its large longitudinal cohort. Data was gathered over time from a substantial group of children, which helps uncover trends that short-term studies can easily miss. The use of both 16S rRNA sequences and shotgun metagenomics allowed for in-depth analysis of both bacterial composition and function, making the findings even more robust. However, there are also limitations that need to be acknowledged. Despite the large sample size, the study population lacked global diversity, which could affect how well the results apply to children in different geographic, ethnic, or environmental backgrounds. To improve this, future research needs to include more diverse populations to see if the same microbial and metabolic patterns are consistent across the world. Additionally, there may be confounding factors that weren't fully accounted for, such as diet, antibiotic use, or genetic differences that could have influenced the microbiome independently of T1D risk. So, while the study is very promising, these limitations remind us that more inclusive and controlled research is needed to fully understand the role of the gut microbiome in type 1 diabetes. As we move forward in our interpretation of the results, it becomes clear that children who developed type 1 diabetes had a distinct gut microbiome profile. Specifically, they show lower microbial diversity and altered bacterial taxa. Recognizing these specific microbial patterns and functional shifts early on may allow them to serve as biomarkers of disease risk. With this kind of insight, clinicians could intervene sooner, potentially delaying or even preventing the onset of type 1 diabetes in at-risk children. What's even more promising is the potential of targeted therapeutic strategies based on these findings. For example, dietary interventions as well as the use of probiotics and prebiotics may help restore a balanced and healthy gut microbiome. While some of these strategies are also used to promote general gut health, the study suggests that they could be intentionally applied to prevent or manage early onset type 1 diabetes. Ultimately, these findings open a door to a future where we can not only predict disease based on the microbiome profiles, but also customize interventions such as nutritional plans or microbiome-based therapies to improve health outcomes. It's an exciting direction that merges microbial genetics with precision medicine, especially for autoimmune diseases like T1D. Before I wrap up the presentation, I'd like to leave you with 5 discussion questions that I think are worth reflecting on and exploring further, especially considering the complexity of the gut microbiome's relationship with type 1 diabetes. How might changes in the gut microbiome contribute to the development of type 1 diabetes? What are the potential mechanisms linking gut microbiota to autoimmune responses in type 1 diabetes? How can this research be translated into clinical practice for early diagnosis and treatment? What are the limitations of this study, and how could they be addressed in future research? What role do diet and environment play in shaping the gut microbiome and its link to type 1 diabetes? These questions are meant to spark thoughtful discussion and help guide for future research, clinical applications, or even your own understanding of microbiome science. I encourage everyone to reflect on them and maybe even dig deeper into one or two areas that really interest you. To wrap up this presentation, I want to highlight the central takeaway. This study underscores the profound role of the gut microbiome in the development of early onset type 1 diabetes. The insights gained not only deepen our understanding of the disease pathogenesis, but also pave the way for new diagnostic tools, therapeutic strategies, and preventative approaches. What's particularly exciting is that this research opens the door to a more personalized and proactive approach in managing and potentially preventing type 1 diabetes, especially in high-risk children. By continuing to investigate how the gut microbiome influences autoimmune diseases, we move closer to transforming clinical care and improving overall public health outcomes. So whether it's through early microbial screening, dietary interventions, or microbiome modulating therapies, this work lays the groundwork for innovations that could truly change lives. Here are my references if you'd like to explore more. Thank you all so much for your attention. I really appreciate you taking the time to listen to my presentation. Have a good one.